Great. Hi. So welcome, everybody. I'm Casper Mossman, Communications Director at QB3. It's a great pleasure to welcome Dr. Aviv Regev from Genentech today. Dr. Regev is Executive Vice President of Genentech Research and Early Development. She manages all aspects of Genentech's drug discovery and drug development activities. By the way, if I'm not getting this right, correct me at any point. Prior to Genentech, she was professor of biology at MIT and an HHMI investigator, and served as chair of faculty and member of the executive leadership team of the Broad Institute of MIT and Harvard. And with Dr. Sarah Teichmann, Dr. Regev co-leads the Human Cell Atlas project to describe all cell types in the human body. And yesterday, I looked this up. I wasn't sure how many cells there were. There's 37 trillion of them, apparently, or thereabouts. That's an awful lot of cells to categorize. Dr. Regev is a leader in deciphering molecular circuits that govern cells, tissues, and organs. Her lab has pioneered experimental and computational methods in single-cell genomics, working toward a greater understanding of the function of cells and tissues in health and disease. She's a member of the National Academy of Sciences and a fellow of the International Society of Computational Biology. Welcome, Dr. Regev. We're going to have a talk for about 35, 40 minutes, and there's a hard stop at 2. So if you've got questions, enter them in the Q&A field, and Dr. Regev will answer them at the end of her talk. Thanks very much.
Thanks for the opportunity to speak here today and for inviting me. And I look forward to talking to you, but especially to hearing your questions and answering them. So I'll start with my disclosures. I'm a co-founder and I have equity in Celsius Therapeutics and in Immunitas. And until July 31st, 2020, I was on the SAB of Thermo Fisher, Neogene, and Syros. On August 1st, 2020, I became an employee of Genentech, which is a member of the Roche group. And I'm going to start with this picture of human genetics and genomics, which you know probably have identified about 100,000 regions in the human genome that are associated with a range of diseases. And while each of these, loci offers some important starting point for understanding the genetic basis of disease and for developing new therapies, at the same time, drug discovery and development remains a really challenging process. And that's true kind of in every step of the way. And so just working through each one clue like that from the genome one at a time is typically a slow and linear process. And so what we need is some kind of approaches that have multiplicative value that would help us to fundamentally shift the pace and the success rate of bringing therapies to patients. And I firmly believe that at this moment in time, there's 4 such big opportunities that I will call my multiplicative levers. The ability to access human biology, or using human as our model organism, the ability to obtain data that has both high resolution and really massive scale, and that helps us match the resolution and scale of biology, or of the molecular space, and so on. And then the third is the ability of algorithms, especially, but not only from modern advanced machine learning, to be able to build models that are predictive, generative, and explainable. And then finally, the diversity of therapeutic modalities that we can develop today. Now, most of my talk today is going to focus on 3 of the levers, and it's going to show how one particular massive scale and high resolution approach, single cell genomics, when applied to human biology and analyzed with modern advanced machine learning algorithms, can give us a roadmap to disease biology. And then at the very end of my talk, and hopefully also in our Q&A, I'm going to try and tie this together with drug discovery and development, which I think of as the missing piece. And that missing piece became very acute for me as a scientist and was the reason that I chose actually to move from the East Coast to the West Coast to head genetic research and early development. So it's a personal, strong feeling for me. Okay. And so with that, I'm going to turn to cells, which are the basic unit of life. Each of us is made of 37.2 trillion cells, give or take. And these cells come in many different types, like neurons and immune cells, muscle cells, fat cells, what have you. Of course, at first glance, we might each think that every cell in our body is the same because it has the same DNA and so the same set of basic instructions. But of course, we all know that the cells are different. They have different structures, locations, they perform different functions in our body. And the reason that this is true is that different cells express different genes. Now, this very basic biology has very critical implications when we think about the genetic causes of disease. And so if there is a genetic variant that confers risk for disease, like those in the lollipops that we saw in the movie in the beginning, all the cells in the body carry the same variant, but disease will really only manifest in those cell types that express this gene. And so knowing our cells is essential in order to understand how the genes that cause disease act. Now, the only problem is that we don't really know how many different cells are there and who they are and what are all of their characteristics. And if you would open Wikipedia, it would tell you that there are maybe 300 major cell types. But of course, if you ask a neuroscientist, they will tell you that just in your retina, there are more than 100 different subtypes of neurons, each of them doing different things. And so what we obviously need is some sort of map or atlas that would have clear and nice coordinates that would let us know all of our cells, what their characteristics are and what they do. But of course, maps do need coordinates. What might these coordinates be? So the idea is that those coordinates can be the level of expression of each gene. Now, because our genome, unlike my cartoon over here, doesn't have only 3 genes, but about 20,000, I can't actually visualize for you a high dimensional space with 20,000 dimensions. But mathematically, that's not a problem at all. And it's also not an issue for algorithms that can discover patterns in high dimensional space and can also show us that in low dimensionality. And you're going to see plenty of plots like that from me today. Okay, so that's also wonderful. But of course, how would we get the data in order to position cells in a map like that? Well, for a long time, we kind of could and we kind of couldn't. We could get high dimensionality genomic data on the levels of expressions of genes, but we couldn't get it for individual cells. And that required new lab methods to emerge. And so very generally, until recently, in order to measure expression profiles, we had to take hundreds of thousands or millions or even billions of cells in a tissue that could be composed of many different cell types, and we would basically measure their average. If you think of every cell as a piece of fruit, we were generating the lab equivalent of a fruit smoothie. In the last few years, there have been major breakthroughs. In single-cell genomics, we can now measure the expression of genes in each cell separately, which we can think of as a fruit salad. And the very latest spatial technologies allow us to do these measurements when the cells are all organized together beautifully in the tissue, which we can think of as the fruit tart. Now, how does single-cell genomics work? It's actually a catch-all to many different types of genomic methods applied to individual cells. I'll focus in this slide and most of the talk on one technique called single-cell RNA-seq. We start with a piece of tissue. We dissociate it to make a suspension of individual cells. We then capture each of the cells separately. We tag and isolate its RNA molecules. There are different techniques to do that. We can then sequence the cDNA actually, and that tells us 2 things: the identity of the genes that they came from and how many copies from them were expressed. And because there are so many cells in the body, we need to be able to do this very quickly and very cheaply for every cell. One approach that allows us to do this is shown in this movie. This is from our method DropSeq from quite a number of years ago now, where the cells get captured in these little droplets in tiny devices. There's other kinds of methods that are also massively parallel methods, and altogether these types of techniques allow us to profile. At first it was thousands and then tens of thousands and now millions of cells that can be readily studied in a day. That of course led to an explosion in the number of cells that we can look at, but also in the volume and noise in the data. And the question is, of course, would we learn meaningful biology from it? And the answer to that question has been algorithms. Algorithms applied to single-cell genomics data really showed us that cellular profiles carry information on many fundamental features of cells: their types and states, their differentiation and other temporal processes, their gene programs, and even the physical location and interactions between cells. And for most of today's talk, what I want to show you is how this information gives us also a new vocabulary for disease studies. in order to address the kinds of questions like where do disease genes act, which cells are disrupted, which programs change in them, how their communications might be affected, and what might be the impact of drug. And so the bulk of my talk today is going to be organized in 3 chapters, starting with cells and then programs and then individual gene functions. I'm going to try and cram as much of it as I can. I might jump through a few things if we run a little short on time. So I'm going to start with cells, and we'll focus on the test case of a common complex disease called ulcerative colitis, which is a form of inflammatory bowel disease, or IBD. And genome-wide association studies, GWAS in human genetics, found hundreds of new risk variants. And these risk variants already pointed to new pathways and processes and could lead to new therapeutics for IBD. But it's also true that in most of the cases, we don't really know the cell of action of the variant or its function or its mechanism. And so in order to try and address this, Chris Smiley and Moshe Bitton, when they were postdocs in my lab, together with Jose Ordovez Montañez, when he was a postdoc in Alex Shalek's lab, and Ramnik Xavier, our colleague at the MGH, assembled a single cell atlas of 365,000 cells that we profiled from 12 healthy donors and 18 patients with ulcerative colitis. For the UC patients, we had both inflamed and uninflamed biopsies, and for the healthy donors, just replicate biopsies of healthy tissue. With the replicate sampling, this leads to about 68 biopsies. All the results I'm showing you are from a training set of about 120,000 cells, and they were nicely replicated in a test set that was collected 2 years later. Now you take all of this data, and we see that the cells partition into 51 subsets, and that includes epithelial cells where we can recover differentiation, stromal cells, and immune cells. And I'm showing you the cells here in these nice spherical embeddings. They're actually embedded on the face of a sphere using a deep learning algorithm that we call SPHERE. And it controls for all sorts of things that make the data difficult to see, like covariates, like patient and sample and disease status, and the channel we ran on, and so on. And now that we have this cell census, we can see that the cellular composition is dramatically remodeled in the tissue as we move from healthy to uninflamed to inflamed tissue. And it includes a whole slew of surprises. So I'll just give you 3 quick examples. If you look at epithelial cells, you see these cells called M cells that are actually very rare in the healthy colon, and they're mostly found in the small intestine in structures called Peyer's patches. But we find that once you look at UC tissue, they're actually greatly increased in their proportions. And that was really unexpected. Then if you look at the stroma, we discovered a new state of fibroblasts, inflammatory state, and that's also a state that's really unique to ulcerative colitis. And then finally, amongst the immune cells, amongst T cells, we see a major expansion of CD8 T cells that express IL-17. That's a very famous cytokine, but it's usually a feature of T helper cells, CD4 Th17 cells. And so now that we have these rich subsets, we can go and ask a very simple question. In which cell is each of these GWAS genes expressed? So we collaborated with Ramnik and Mark Daly in order to look at loci that they had already mapped to the individual gene level. And more than 50% of those genes were expressed in specific cell subsets, either irrespective of disease, that means all, or only in healthy or only in inflamed tissue. And now we can turn back to the cells and we can search for cell subsets that are enriched for GWAS genes. So for example, these M-like cells that I told you are uniquely occurring in ulcerative colitis, they also have a preponderance of GWAS genes that they specifically express. And moreover, this expression is induced in the M cells in ulcerative colitis compared to M cells, the very few M cells that you have in the normal colon. Now we asked, how are the cells that are highlighted by this analysis might relate to each other in the tissue? So we built a graph that connects cells that are enriched for corresponding receptor ligand interactions in each of the 3 conditions. So in the healthy tissue, you see that the key compartments, epithelia, stroma, immune, are mostly separated from one another. Cells interact with each other within a compartment, say immune cells with immune cells, but not between the compartments. But in ulcerative colitis tissue, in uninflamed and even more so in inflamed, they're actually decompartmentalized. We see a lot of cross-compartment interactions. And some of the cells that were enriched for expression of these GWAS genes, they are the key hubs of this compartmentalization. For example, those inflammatory fibroblasts, the M cells, Tregs, and others. Okay. So now let's go back to the GWAS genes and ask, but how well have we actually accounted for them? So as you can see, a little over a half had cell type specific expression in our colon data, but some of the others don't even seem to show any expression in our data set. So what's happening with them? So there could be multiple reasons for that, but one of the major ones is that we do not have a full picture of the tissue for two different reasons. The first one is that we only profiled the mucosa and the lamina propria, which are the outermost layers, the ones that face most into the gut lumen, but some cells like adipocytes exist only in other layers, like the muscularis layer at the bottom. Second, there's a few cell types that we didn't capture at all. For example, enteric neurons. These are neurons whose cell bodies live in the gut. We know that they're there. And it's not just that enteric neurons are really rare cells, but also neurons do not survive dissociation for single cell RNA-seq. And so we decided to tackle both of these challenges by developing two new lab methods, RAISIN-seq, that allows us to profile nuclei from many different tissues, including the gut, and Miracle-seq for label-free enrichment of rare cells. And so Jean Brokliansky, when he was postdoc in the lab, together with Chris Smiley and Nick van Wittenberger, combined these methods and applied them to frozen resections from another 10 individuals in order to build an atlas that gives us those deeper layers. And they recovered very rare cell types, not just enteric neurons, but even interstitial cells of Cajal that are even rarer than the enteric neurons, as well as other cell types that were hard to recover, like adipocytes. And now we can go back with these additional cell types to the UC GWAS genes, and we can see that we can basically relate every GWAS gene to a cell, including to very specific subsets of enteric neurons, but also to cells like adipocytes. Now, once we have this, we can also identify cell type specific expression of dozens of risk genes for disorders that affect the enteric nervous system in particular, like Hirschsprung's disease, and also early life and late onset CNS disorders like autism spectrum disorders and Parkinson's disease, all of which are known to have concomitant phenotypes in the gut, like gut dysmotility, that the gut doesn't, contract well. Now, in some cases, the expression in enteric neurons is a lot higher than in CNS neurons. And that suggests that the gut in those cases can be a key locus of the action of the gene. And also that we could use the gut as an accessible source of tissue in patients for some of these diseases. So this kind of example emphasizes how important it is for catalogs to be comprehensive. And in fact, these kinds of methods that we originally developed for the ENS also allowed us to build an atlas of COVID-19 autopsies from the lung, the heart, the liver, and the kidney. I'll come back to this with a little vignette a little later on. We recently published this, as well as a cross-tissue atlas across 8 tissues from which we deciphered cell and tissue-specific programs for parenchymal, immune, and stromal cells. So to summarize this first part, I showed you a glimpse of what a cell atlas looks like. This one was built for ulcerative colitis, a complex common disease, and we built it from involved and uninvolved tissue in patients and compared it to a healthy reference from healthy individuals. A relatively small number of individuals gave us a very robust catalog. Most of the GWAS genes were expressed in a specific cell subset. And then as we expanded the tissue coverage from the mucosa to the inner layers and captured the rel cells with the new lab methods, we're able to relate nearly every gene to a cell type. And now we're expanding this in the context of multi-organ atlases, which we did both for COVID-19 and for healthy tissue. Okay, so until now, things were really simple. We had like this basic unit of the cell, and we looked at everything from the perspective of the cell, like in which cell is a gene expressed? Which cell types do I have? But can we look from a different perspective, the perspective of the genes, and start thinking about how the genes relate to each other in programs? So to motivate this, let me take you back to the analysis in ulcerative colitis and to a gene with the name C1orf106. It's a gene associated with ulcerative colitis risk based on genome-wide association. But the function of the gene was not known at the time, which is really indicated by the uninspiring name. It says that it's on chromosome 1. What we do know from the atlas is that it's expressed in enterocytes. And one way of predicting the function of genes is asking which other genes are they expressed with. It's called guilt by association. And so in the past, if you wanted to do this, we would compare one cell type's expression to another cell type's expression. And we can do this by looking at the covariation of C1orf106 with other genes across all the cells in the atlas. And what you would get are genes like EpCAM and keratins, because this gene is expressed in enterocytes. Enterocytes are epithelial cells. This is an epithelial cell signature. But with single-cell RNA-seq, we can do better because we can look at the subtler variation in C1orf106 just across the single enterocytes. And we find that it co-varies with tight junction genes. So we can postulate that it has a function in the tight junction and the barrier in the colon. And indeed, Ramnick's group showed in a separate study that it regulates the stability of epithelial adherence junctions. So we said, bingo, let's generalize this idea across all the GWAS genes. Let's take a GWAS gene, relate it into cell types, look for genes that are co-varying with it within the same cell type, get ourselves a process. And when we started doing this, we saw that often other GWAS genes were coming along for the ride. Say, I start with C1orf106, I also get gasdermin B, GPR35, and SMAD3, not just the tight junction genes. So we decided to iterate the process, starting with each different GWAS gene as a seed, until we would converge to a set of modules that minimally connects the GWAS genes and additional ones. And remarkably, most of the modules that we get are enriched for multiple GWAS genes at a time, and 10 such modules together span more than half of the UC GWAS genes in a cell type specific manner. So now we said, well, if we know this structure exists, can we use it in order to nominate risk genes in loci that have multiple genes in them? That means if one characteristic of GWAS genes is that they aggregate together in modules, can we prefer candidates that co-vary with known GWAS genes or with candidates in other regions? So to test this kind of idea, we took cases where the causal gene is known in a multi-gene region. We hid its identity, and we saw if using either expression or covariation with genes in other associated regions helps us predict the causal gene. And we find an almost twofold increased accuracy compared to a null model where we choose the gene at random or by its expression alone. And this was true only in ulcerative colitis, which is the source of our tissue, but not in Crohn's disease. And now we can take regions that are not known and predict new causal genes, and these are just new hypotheses for your geneticist friends. Now, this kind of map of cells and gene programs allows us also to compare better between related but different diseases. So in the case of IBD, in addition to the common form of IBD, you have many genes and each with common variants that contribute a little bit to disease risk. There's also monogenic forms of the disease where you would have one gene with a major genetic defect that would usually lead to more severe disease earlier in life, sometimes in extremely young children and babies. And so in a collaborative study with Holm Ulig in Oxford, Chrissy Bolton in Holm's lab, together with Chris in my lab, tried to put these two perspectives together. So in some cases, the defects are simply in the same genes. If you take the 81 monogenic IBD genes and 278 polygenic IBD genes, 13 are simply the same gene, just a different level of defect. But if we ask in which cells are the genes expressed and in which programs, we have a much higher overlap. So even when it's in different genes, we can see that the different diseases actually converge to the same cells and the same gene programs. Now, most recently, we took this a step further, and Karthik Jagadish, who's a postdoc in the lab, together with Kushal Dey in Alkes Price's lab, built a framework to relate gene programs from single-cell RNA-seq datasets, GWAS summary statistics, and enhancer gene linking strategies using a new single-cell LD score regression framework that we called SC-Linker. And SC-Linker gives us basically a chain from the disease to a program that's related to a cell type. We actually analyzed 1.6 million cell profiles, 209 individuals, 11 tissue types, and 6 disease conditions, together with GWAS and PHWAS from 60 diseases and complex traits. And the answer that we get are of the form that I'm showing you here. We used 3 different kinds of programs. With cell type specific programs, we showed, for example, how part of the heritability for Alzheimer's disease is explained by monocyte programs, and that the MS4A6A gene contributes to this. With disease progression programs, we see that genes like FERMT1 that are differentially expressed in M cells from ulcerative colitis versus healthy, they explain UC heritability. And with pathway-related programs, that can be, a program active varies within one cell or active in multiple cell types. We see, for example, that the complement cascade program explains the heritability in MS, including through a gene CD37. Now, you can apply this to anything. So, for example, we used it in the context of COVID-19 in order to suggest cell types that remediate the heritability of risk of developing severe COVID-19 disease. And by combining our single cell data and the GWAS for COVID-19, we could nominate 82 cells, ciliated cells, and CD8 T cells in the lung, cholangiocytes in the liver, and connected and proximal tubule cells in the kidney. Okay, so up to this point, we looked at programs one cell type at a time. But if I think from a tissue biology perspective, which is where disease actually happens, we expect the cells to coordinate their action, either because they're all responding to a shared signal, or because one of the cells responds to the signal and then cues the other cells around it. And they can respond in the same way or in different ways. And that's the 4 quadrants in this image. But regardless of which of these 4 scenarios happens, it means that we would expect a signal in the data that reflects this coordination. And I will call this signal multicellular programs. It can be completely different genes in different cell types, but they should be going up and down together, given the condition of the tissue. And so in order to relate multicellular programs and find this kind of signal, we need some way to relate the cells to each other. Spatial proximity is one obvious way, but I'm going to show you that it can also be looser than that, like tissue locations or individuals and disease conditions. And so to look for these kinds of signals, Livnat Jerby, when she was a postdoc in the lab, developed a 2-step approach. She's actually local in the Bay Area now, in Stanford. And she called this approach dialogue. And in dialogue, we learn a new latent feature space where the cell type specific representations have dependencies between different cell types across either different physical niches in spatial data or samples or conditions in single cell data. And that's where the multicellular programs come from. So here's an example of this in spatial data. In this case, it's the mouse hypothalamus. And just to illustrate it, I'm showing you the cells involved in 2 multicellular programs, MCP2 and 4. In each case, the cells are excitatory neurons in red and inhibitory neurons in blue, but different cells in different spatial locations have coordinated expression of different genes. And that gives the 2 different spatial patterns. Now we wanted to take this idea and apply it to profiles from dissociated tissue. So we no longer know which cell is immediately next to each other, but we do know that they came from the same sample, for example, from the same little biopsy, in the UC atlas that I showed you. And in fact, we had 68 biopsies there, but we actually had 115 spatially distinct samples because for each biopsy, we separated the lamina propria and the mucosa layer before we did single-cell RNA-seq. And we focused not on 2-way patterns, just one cell type and another, but up to 5-way patterns for the most well-represented subsets in the data set: macrophages, 2 subsets of transit amplifying intestinal epithelial cells, CD8 T cells, and CD4 T cells. And here is just one example of a multicellular program. The 5 different cell types are labeled on the top. The genes are different in each of the cell types, but the levels of these genes in the cells are coordinated across the cells from the 5 types across the 115 samples. And this program is induced across these cells in ulcerative colitis, and it's enriched in multiple genes located in risk loci for IBD and UC. So even though these GWAS genes are expressed in different cells, their expression is coordinated across these cells altogether. And then finally, we can score this program in bulk patient RNA-seq, and it's predictive of responses to anti-TNF therapy. And so to summarize this second part, you see GWAS genes fall into co-varying modules with each other and with other genes. And as a result, more than 50% of the GWAS genes for UC mapped into these 10 meta-modules based on the mucosal atlas alone. And then the modules helped us predict GWAS gene function and improve our prediction of causal genes in multi-gene regions. And then we incorporated this module structure in order to compare polygenic and monogenic IBD, and also in order to relate cell types to traits using SC-linker, with these programs that reflect cell type and disease progression by cell type and cross-cell type programs. And then finally, I showed you a first attempt to recover multicellular programs across multiple cell types in one tissue. Okay, so now for the third and last chapter, I'm going to ask whether these approaches can help us also decipher gene function. And we want to do this by looking directly at genetics as a source of perturbation or intervention in order to get to causality. And we can do this relying on natural genetic variation or by introducing perturbations in an engineered way. And in both of these cases, we'll rely on the single cell profile as our high content readout. So to begin doing this for engineered perturbations, a few years ago, we developed Perturb-seq in order to combine a pooled CRISPR screen with single cell RNA-seq as the readout. And after computational analysis, you recover a regulation matrix like this one here from our early screen in dendritic cells that were treated with LPS and perturbing 24 transcription factors. And you can see how the perturbed transcription factors in the columns group into these co-functional modules of factors that when we perturb any of these factors lead to similar impacts. And the profile genes partition into programs of genes that are affected in similar ways across the perturbations. And all of this is in ways that recovered good dendritic cell biology, as well as some novel discoveries. And so what's nice about these Perturb-seq screens is that just like cell atlases, they're unifying. You can take one readout and apply to understand effects in vivo or in vitro, in coding or non-coding variants for one cell type or for multiple ones at once. And you can use perturbations that are genetic, but you can also use small molecules and other types of interventions. So I'll give you a few very brief examples on this. I know our time is running on the shorter end. So for example, Xin Jin, when she was a junior fellow in Fang Zhang and Paula Arlota's lab in collaboration with Sean Simons and Joshua Levine in our group, used Perturb-seq in order to characterize the function of 35 genes with large effect loss of function variants in autism. So here we know that each of these genes is causal for disease, but we don't know, again, in which cell or in which process it participates. And so we set up a screen where we deliver a pool of guides to mouse embryos in utero, and then the cells are recovered and profiled postnatally. And when we characterize the effect of perturbations on major cell types, only one of the 35 genes, DIRC1A, had a significant effect on the proportion of major cell types. It increased oligodendrocyte cells and reduced microglia. But when we turn to the level of programs, we got something different. So we first identified gene programs across the entire data set, two different types of patterns. I'm showing them in projection neurons. So for example, some programs like PN1, they're really subtype diversity. Here it's layer 4 projection neurons. You would have gotten this from clustering. But other programs like PN2 are used across the subset. And so they highlight a subset of the cells within each type. And this is sensible here because the process is neurit development that all neurons have to go through. And so here, when we look at the level of programs, 15 of the 35 ASD genes affect 6 of these programs across 4 cell types. And this really highlights the likely limited number of cellular processes that the genes converge into. And also that a perturbation in one gene can impact programs in multiple cell types. So for example, ANC2 induces the NDNF module in inhibitory neurons, but it also affects modules in astrocytes and in projection neurons. And SHID8 and GATA2B both alter the oligodendrocyte 1 module, and they affect oligodendrocyte progenitor differentiation, and we validated that in individual experiments. And so naturally, these kinds of results are in the mouse, and ASD and other neural disorders are actually notoriously difficult to study in animal models. So the question is, can we get closer to human by leveraging something like, say, brain organoids? And so here ensued an extensive series of collaborations with Joshua Levine and Paula Arlota to really achieve this goal, and I'll blitz through them. So first, Silvia Velasco, when she was in Paula's lab, led an effort to set reproducible brain organoid models, and we used single-cell RNA-seq of almost 500,000 cells across 69 organoids, made in 20 batches from 5 different cell lines in order to show a high level of reproducibility. Then Amanda Kedaigle and Anna Uzquiano focused on corticogenesis. And they followed a dedicated 7 time point time course to show again that organoids are reproducible and they generate the right molecular signatures across different neuronal subclasses. Now that we had those, Bruna Paulsen and Marina Pignon, together with Sylvia and Amanda, turned to do Perturbation, looking at 3 haploinsufficient mutations in SUV420H1, ARID1B, and SHID8. All in our original set, were in the original set of the ASD loss of function genes that we assayed in the mouse, but now we're in human organoids. And in each case, we profiled enough cells from at least 24 organoids, 2 to 3 cell lines, multiple batches, 2 time points. And we found that all 3 haploinsufficients converge on a joint phenotype of accelerated development of GABAergic neurons. And in addition, the ARID1B haploinsufficiency led to delayed generation of deep layer projection neurons. And you can see this in the difference in the inferred pseudotime distributions compared to control at the bottom. And on the other hand, the SUV420H1 haploinsufficiency led to an accelerated generation of deep layer projection neurons, and with the opposite pattern in the inferred pseudotime distributions. And then, and really interestingly, the penetrance of the GABAergic neuron phenotype in SUV420H1 mutations dependent on the genetic context, meaning the source of the organoids. And that suggests that there's additional modifiers that can interact with this mutation. And so when we take all of these pieces together, we see that mutations in 3 of the top genes associated with autism, SHID8, ARID1B, and SUV420H1, converge on abnormalities in 2 populations of neurons, the GABAergic neurons and the deep layer projection neurons. And also in both populations, the mutations impact the speed of development of these 2 populations. For GABAergic neurons, it's in the same direction. For deep layer projection neurons, sometimes in opposite directions. Sometimes there's genetic modification of all of those things. And I didn't have time to show you, but there's additional evidence from proteomics that these are shared phenotypes, but they happen largely through distinct molecular pathways. And moreover, that we speculate that this all has to do with abnormal activity of local circuits. Now, I'm going to actually skip the next 3 slides. This was a tiny little vignette on discovering anti-COVID antibodies using single-cell genomics. It's published and you can look at it. But I do want to make the point that these were all studies in the dish in one way or another. It can be the organoid dish, it can be the cells, or in an animal model. But we really also want to understand, and they were all engineered. But can we also use these kinds of approaches to look at the impact of natural genetic germline variation? And while the example I'm showing is in the mouse, this was our practice ground towards the human. So in this example, we're back in the gut. We're looking at cells called innate lymphoid cells, or ILCs, in the small intestine. We profiled 380,000 cells across 274 diversity outbred mice, so to get as close as we could for human genetics, and we used pooling and genetic demultiplexing to make the experiment feasible. And now we can map genetically different kinds of traits. We can look at EQTLs, heritable variation in gene expression at the level of each cell type, but we can also look at cell proportions as a trait, and even trans association by looking at the impact on gene programs. And that's the vignette that I'm going to highlight. This is a program that varies across 2 subsets of cells, ILC3s and LTIs, and it is enriched for genes from the IL-22 pathway. Now, IL-22 is produced by ILCs and then signals to enterocytes, and it's very important for gut physiology and maintenance. Now we go to the genetic side, we map loci, we get 3 loci. One of the 3 harbors a very famous transcription factor, SOX9, except that SOX9 is a transcription factor that's expressed in enterocytes, not in ILCs. So this suggests that there's a feedback loop between ILCs and enterocytes, genetic variation that first impacts SOX9 locus, that affects the enterocytes, that is sensed by the ILCs, that generate IL-22, which comes back and impacts the enterocytes. And we found actually multiple examples like this, including the QTL and CGRP, which is a gene expressed by enteric neurons that impacts ILC2s, and that's actually a well-established axis in the gut from our own earlier studies. And so these might look like really big experiments, but actually we'd love them to be much bigger, to genome scale, small molecule screening, beyond. So power analysis of these earlier Perturb-seq studies showed us that if we want to decipher program level regulation, 30 to 50 cells per perturbation should suffice. So a genome-wide screen means about 3 million cells profiled. That's totally doable. It's so doable that it has already been done. But I want to say there's also clever approaches on how to do this more efficiently. And one that we have tried to develop, we call Perturb-Me, that combines enrichment of cells with a desired phenotype using flow-based fluorescent antibodies, fluorescent labeled antibodies, with single-cell RNA-seq readout for the protein and the RNA level. And again, because of the time, I'm going to actually skip the coming few slides on this specific topic. So to summarize this third part, Perturb-seq is a family of methods that couples pooled perturbation with high-content readout. Our original application showed it for CRISPR loss-of-function screens with single-cell RNA-seq readout. It can be naturally modulated for one or multiple perturbations per cell. It can be coupled with other readouts like attack or even cell images in pooled optical screens, as my good friend Paul Blaney showed. I showed you examples of using it in vivo in a screen of loss-of-function variants in ASD. I showed you it in organoids. I showed you a version with natural genetic variation. I will tell you, and we've published examples of this in co-culture, protein, mRNA, coding variants in cancer, and there's more and more coming up. And we have worked now approaches to scale this to the millions of cells and different ways of tackling combinations of perturbations. So with this, I'm going to conclude these 3 chapters. I've showed you something about the power of atlases to already impact many aspects of our understanding and treatment of disease, including cancer, human genetics of disease, regenerative medicine, and more. And so that brings me to where I left you off in the very beginning. Atlases are kind of this great live demo of the power of putting together human biology measured by high resolution and massive scale methods to generate data from which we can decipher all aspects of disease using machine learning and other algorithms. But knowledge of disease is not the same as treating disease. And for me, in choosing to come to Genentech, I really sought to combine these 3 levers with the 4th and very important one, the ability to discover and develop medicines with diverse therapeutic modalities. And so in closing, I wanted to give you a 1-minute glimpse of how we're trying to build this multiplicative future of a drug discovery and development portfolio, where our ultimate goal, as always, is to treat, restore, and cure. So today I showed you a lot of the power of combining human biology and high-resolution massive scale approaches. To bring those to the next level, and especially in the context of disease, we set up a new department for cell and tissue genomics and 2 large initiatives, translational genomics, which pushes, from discovery to the clinic, and reverse translation that starts actually with the patient in clinical trials and goes back to discovery. Now, there are specific areas of biology and disease areas that are really ripe for these kinds of advances, including screening complex cell biology phenotypes, molecularly and by imaging, targeting cell programs and states, finding new ways to program cells, either as a cell therapy or in the native tissue, deciphering tissue biology in diseases, looking beyond cells to how cells relate and affect each other through multicellular programs, all that stuff. And just like the examples that I showed you today, each of these programs requires the combination of human biology, massively parallel high-resolution methods, and computation. And each of them is rapidly uncovering new biology, so new target spaces for us. But beyond biology, there is much more. So for example, we're also growing and applying the same kinds of strategy that combine rich data and large models in order to generate new molecules. And that is by establishing what we call a lab in a loop. So in each of these cases and others, we start with large-scale data, for example, from antibody engineering experiments, from small molecule screens, from antigen presentation. And then we use it to train new predictive models. And then we use the trained models to make new predictions, which we then feed back for more experiments. And this lab is a loop is basically what you saw in all those cell perturbation screens that I showed you, except that here it's applied for molecule discoveries. And in all of these cases, the space of possibility is far greater than the atoms in the universe, but the computational models trained on large data can still transverse and make great predictions. And then, of course, there's many more loops like that. There are great loops similarly to that in the clinic. I'm going to stop here. I want to leave us at least 15 minutes for the questions that I will be very, very happy to take.